Today, we will be demonstrating a method called Thin Layer Chromatography, or TLC, which can be used to identify and detect the presence of ethylene glycol or diethylene glycol in cough syrups. Chromatography is a technique typically used to separate and identify different components within a mixture. As we can see here, the various components from a drop of ink are separated into different colors using this technique. We would like to utilize this separation method for the detection of ethylene glycol in cough syrup samples. To do this, we would require the following materials: potassium permanganate staining solution, a TLC chamber, some cough syrup, different samples for analysis, a TLC plate, forceps, a pencil, capillaries, and a hairdryer. Demonstration of this TLC procedure is performed by our final year undergraduate student, Julian. To achieve this, firstly, insert the capillary into the sample to collect some liquid. Next, briefly and gently tap the end of the capillary onto the TLC plate so that some of the sample is spotted on the plate. Repeat these steps until all the samples are spotted on the plate. This is how our plate is set up. Our first two lanes consist of pure ethylene glycol and diethylene glycol, which are standards. Cough syrup A is in the middle lane, and the last two lanes comprise of the cough syrup spiked with ethylene glycol or diethylene glycol with respective concentrations shown. After spotting the plate with the samples, use a hairdryer to ensure that the TLC plate is dried completely. Then, the TLC solvent, made up of the following components, should be poured into the TLC chamber. Swirl the chamber thoroughly to ensure good mixing. Next, using the forceps, gently place the TLC plate into the chamber. Allow the solvent to rise on the TLC plate by capillary action. This should take about 2 to 3 minutes. Once the solvent has reached the top, cautiously remove the TLC plate and dip it into the staining solution after partial drying in the air. Ensure that the complete plate is submerged. To visualize the samples that are on the TLC plate, simply place the TLC plate on a hot plate for drying, and in a few minutes, you will observe that the plate is dried completely. This should reveal the different components that are present in our samples. In the example shown here, spots of pure ethylene glycol or diethylene glycol appear on the TLC plate at about the same distance from the baseline. The unspiked cough syrup shows 2 spots, but the spiked cough syrup samples clearly indicate the presence of ethylene glycol or diethylene glycol. This is an extremely sensitive detection tool and also works on samples that are spiked with as low as 0.05% of ethylene or diethylene glycol. However, at high concentrations of 10%, the spots smudge and may lead to difficulties in interpreting the results. It is best to dilute the samples should one encounter this problem. Lastly, this method also works on cough syrup B. We would like to express our sincere gratitude and extend our heartfelt thanks to the entire team for their continued effort, which has played an important role in making this project a success.